Namaskar, hello and a very warm welcome to this special webinar series of ours, Listening to Learn. My name is Tanvi Khurana. At this moment, आप हमें देख रहे हैं ई-विद्या चैनल नंबर 9, 10, 11 और 12 पर। आज हम आपके लिए जो विषय लेकर आए हैं दैट इज़ एक्सट्रीमली इम्पॉर्टेंट, क्योंकि वो आजकल बहुत ज़्यादा हाई डिमांड में है, पर उससे जुड़े बहुत सारे सवाल हैं जो हम अपने एक्सपर्ट से पूछना चाहेंगे। आज का टॉपिक है बायो इन्फॉर्मेटिक्स, डेवलपमेंट एंड फ्यूचर, जिसे हम हिंदी में कहेंगे जैव सूचना विज्ञान, विकास एवं आगे की राह। अगर आपके पास कोई सवाल है, कुछ पूछना चाहते हैं, कुछ भी जानना चाहते हैं आज के इस विषय से जुड़े हुए, तो आप हमसे बातचीत कर सकते हैं। नो मैटर वेयर यू आर एट दिस मोमेंट, प्लीज वॉच अस ऑन आवर यूट्यूब चैनल दैट इज NCERT ऑफिशियल। इन द लाइव चैट बॉक्स यू कैन रेज योर क्वेश्चंस, योर क्वेरीज एंड यू कैन शेयर देम विद अस। अगर आप चाहें तो हमें फ़ोन भी कर सकते हैं। नंबर है 8800440559। बहुत सारे माध्यम हैं जिनके ज़रिए आप हम तक पहुंच सकते हैं, अपने सवाल हम तक पहुंचा सकते हैं। दिस इज़ नॉट एन ऑर्डिनरी प्रोग्राम, क्योंकि हम हर रोज़ यहाँ आपसे मुलाकात नहीं करते हैं। हम एक महीने में कोई भी 2 फ्राइडेज़ को आपसे मिलते हैं और एक ऐसा टॉपिक लेकर आते हैं जो आपके लिए इम्पॉर्टेंट होता है, जो सबके लिए इम्पॉर्टेंट होता है। बायोइन्फॉर्मेटिक्स आज हम यहाँ डिस्कस करेंगे और मेरे पास हैं बहुत सारे सवाल, पर आप सभी को बताना चाहूँगी इट्स बिन मोर देन 2 years सिंस वी स्टार्टेड दिस प्रोग्राम एंड थैंक यू टू ऑल ऑफ़ यू फॉर मेकिंग इट सो सक्सेसफुल। हमने आज तक बहुत सारे टॉपिक कवर किए हैं एंड वी हैव गॉट ग्रेट रिस्पॉन्स फॉर दैट। आई रिक्वेस्ट यू, अगर आपके पास कोई भी ऐसा विषय है जो आप चाहते हैं हम इस प्लेटफॉर्म पे डिस्कस करें, तो प्लीज़ हम तक ज़रूर पहुंचाइएगा। डिपार्टमेंट ऑफ एजुकेशन इन साइंस एंड मैथमेटिक्स, दैट इज़ DESM, NCERT न्यू दिल्ली ब्रिंग्स यू दिस नेशनल स्टूडेंट आउटरीच प्रोग्राम। आज जो सेशन है वो है हमारा 67th सेशन। जी हाँ, 67th सेशन है हमारा, और जो हमने पास टॉपिक्स डिस्कस की हैं उसमें बहुत कुछ हमने कवर किया है, चाहे वो मुंबई डब्बा वाला हो, चाहे वो बैटरी मैन्युफैक्चरिंग हो या स्पेस से रिलेटेड हो, बहुत सारे टॉपिक्स हमने कवर किए हैं। और मैं इतनी बात नहीं करूंगी क्योंकि हमारे साथ मौजूद है हमारे स्टूडियो में द ब्रेन ऑफ दिस एंटायर प्रोग्राम एंड ही इज़ डॉक्टर गगन गुप्ता। सर, अ वेरी वार्म वेलकम टू यू।
थैंक यू तन्वी। नमस्कार दोस्तों, सादर प्रणाम। और आज का प्रोग्राम हमारे लिए बड़ा इम्पोर्टेंट है। एक बड़ा इंटरडिसिप्लिनरी विषय पर आज हम बात करने वाले हैं, जैव सूचना विज्ञान। इस पर आज हम बात करने वाले हैं। दोस्तों, ज़रा जैव सूचना विज्ञान पर 2 मिनट में आता हूँ आपके पास। आज हमारा 67 सेशन है। थोड़ा सा मैं आपको पीछे की तरफ ले चलता हूँ। जब अगस्त 2021 में जब हमने इस प्रोग्राम को, इस कार्यक्रम की शुरुआत की थी, तब हमारे साथ इस प्रोग्राम में नेशनल ह्यूमन राइट कमीशन और काउंसिल ऑफ साइंटिफिक एंड इंडस्ट्रियल रिसर्च CSIR के एक यूनिट हमारे साथ इस प्रोग्राम में जुड़ी हुई हैं, अभी तक जुड़ी हुई हैं, और उनके साथ हमारा प्रोग्राम शुरू हुआ था। हमने नहीं सोचा था कि हम कितना आगे तक बढ़ेंगे। जैसा मेरी साथी तन्वी ने अभी कहा कि आप लोगों के प्यार से, आप लोगों की मोहब्बत से हम लोग आगे बढ़ रहे हैं, और ये प्रोग्राम, और आपने हमें इसे सक्सेस दिलाई है। इस प्रोग्राम को सब काफ़ी सक्सेसफुल बनाया है, और हमारे शेड्यूल्स काफ़ी आगे तक तय हो चुके हैं, और हम इस प्रोग्राम को नियत कर रहे हैं। आपके जो ये इसलिए संभव हो पाया है क्योंकि हमें आप लोगों से बहुत फीडबैक मिलता है। आप लोगों हमें सुझाते हैं कि किस तरह के टॉपिक्स को लें, और उस तरह के टॉपिक्स को हम लेते रहे हैं, चाहे वो डिफेंस से रिलेटेड हो, चाहे वो स्पेस से रिलेटेड हो, चाहे वो मेडिसिन से रिलेटेड हो, चाहे वो मेडिकल रिसर्च और साइंटिफिक रिसर्च से रिलेटेड हो, चाहे वो मैथमेटिक्स से रिलेटेड हो, मैथमेटिकल एजुकेशन से रिलेटेड हो, मैथमेटिकल साइंस डेवलपमेंट से रिलेटेड हो, मॉडलिंग से रिलेटेड हो। आप ये सारी लिस्ट आप हमारे ncert.nic.in, जो कि हमारे NCERT की ऑफिशियल वेबसाइट है, इसके इवेंट सेक्शन में देख सकते हैं। ये सारे सेशंस और सारे 66 सेशन जो अभी तक हुए हैं आपको उस सेशन में मिल जाएंगे। आप वहाँ से इन सबको डाउनलोड भी कर सकते हैं, देख सकते हैं। आज का सेशन भी आज के सेशन के बाद आपको उस सेशन पर मिल जाएगा। मेरे साथ हैं आज मेरे दोस्त डॉक्टर नरेंद्र, एक युवा साथी मेरे, और आज क्योंकि युवा साथी हमने इसमें इस विषय पर लिया है क्योंकि ये सब्जेक्ट भी थोड़ा युवा है, नया है हम लोगों के लिए, सभी के लिए। और आज इस के लिए जो डॉक्टर नरेंद्र दिल्ली यूनिवर्सिटी में है, मैं इनका तारुफ आपके साथ कराऊंगा अभी एक थोड़ी सी देर में। दोस्तों, बायो इन्फॉर्मेटिक्स ये जो एरिया है, ये इसका वर्ड का कॉइन लगभग 1930-40 के आसपास हुआ था। डॉक्टर साहब हमें थोड़ा सा समझाएंगे अगर समय मिला। उसके बाद डीएनए रिसर्च हुआ, डीएनए डिस्कवर हुआ 1950s अर्ली 50s में। और उसके बाद में जब थोड़ा बहुत इसके ऊपर इस एरिया में जैव सूचना जिस तरह से नाम है, बायोलॉजिकल सिस्टम्स के बारे में इंफॉर्मेशन कैसे मिले, किस तरह से हम उसके साथ कॉम्पीट कर पाएँ, इस सब के बारे में हमने बात करना शुरू किया। इस पर धीरे धीरे रिसर्च होती रही और फिर धीरे धीरे 1970s का दौर आया, जबकि कंप्यूटर्स की जो जैव सूचनाएं हैं उसको संगणक और उसके कैलकुलेशन और उसके बारे में प्रेडिक्शंस करने के, प्रोसेस करने के लिए हमारे पास अवेलेबल थे। साथ साथ मैकेनिकल कंप्यूटर से इलेक्ट्रिकल कंप्यूटर्स और मेन फ्रेम कंप्यूटर्स पर आ चुके थे 1970s में। और आपको दोस्तों याद होगा कि इसके बारे में हमने बहुत बात की है, कंप्यूटर्स के एवोल्यूशन के बारे में। और आपको फरवरी 2022 में राजकुमार भैया हमारे साथ ऑस्ट्रेलिया से जुड़े थे, मेलबर्न से। उनके सेशन को आप देख सकते हैं। आप उस सेशन में आप पाएंगे कि कंप्यूटर्स किस तरह से इवॉल्व हुए। अभी भविष्य में, अभी फरवरी के महीने में अब हमारे साथ रामा राव यूएसए से जुड़ेंगे, जो कि शायद हम कोशिश कर रहे हैं कि वो हमारे साथ स्टूडियो में आ जाएं दिल्ली में ही। तो आप उनको बात सुनेंगे, जो कि फिर से एक बार पुनः कंप्यूटर किस तरह से इवॉल्व हुए और कंप्यूटर के प्रोग्राम्स और प्रोग्रामिंग लैंग्वेजेस, प्लेटफॉर्म्स किस तरह से डेवलप हुए हैं, उसकी पूरी जेनेसिस हमें यहाँ से समझाएंगे लेकिन आज मैं उस सब का जिक्र इसलिए कर रहा हूँ क्योंकि कंप्यूटर्स की जब विकास हुआ, कंप्यूटर के प्रोसेसिंग का विकास हुआ, प्रोसेसिंग टेक्निक्स का विकास हुआ, तो 90 के दशक में जब सुपर कंप्यूटर्स आ चुके थे और प्रोसेसिंग जिसको कि आज हम विद इन कोट मार्क को प्रोसेसिंग कहते हैं, वो जब हमारे सामने आ चुका था, तब बायोलॉजिकल सिस्टम्स की इंफॉर्मेशन को लेने के लिए एक ये बायो इन्फॉर्मेटिक, जो कि थोड़ा पुराना, इतना नया नहीं था, लेकिन उसके बारे में लोगों को समझ थी और उसके रिसर्च में और उस एरिया की एक्टिविटीज़ में काफ़ी बूम आया, काफ़ी प्रगति हुई और वो प्रगति आज तक चल रही है। और ख़ास तौर से मेडिसिन के एरिया में इस सब के बारे में बात हुई और ये एरिया काफ़ी प्रोग्रेसिव एरिया है रिसर्च का। दोस्तों आज मेरा आपसे अनुरोध है कि आप जो स्टूडेंट्स हैं और जो यंग रिसर्चर्स हैं वो इस टॉक को बड़ा ध्यान से सुनें और आपको हो सकता है कि आपको आगे आने वाले कैरियर या अपने फ्यूचर रिसर्च के बारे में कुछ आइडियाज़ मिलें, कुछ डायरेक्शंस मिलें, क्योंकि ये एक ज़बरदस्त मल्टी डिसिप्लिनरी, इंटरडिसिप्लिनरी एरिया है जिसमें फिजिक्स के लोगों की, केमिस्ट्री के लोगों की, बायोलॉजी के लोगों की, मेडिसिन के लोगों की, सब एरियाज़ के लोगों की ज़रूरत है, इलेक्ट्रॉनिक्स के लोगों की, इलेक्ट्रिकल साइंस के लोगों की ज़रूरत है इस एरिया में। तो इस एरिया में काफ़ी स्कोप है अभी रिसर्च करने के लिए। इससे पहले कि मैं अपने साथी डॉक्टर नरेंद्र के से आप रिक्वेस्ट करूं कि वो आपसे बात शुरू करें, मैं डॉक्टर नरेंद्र के बारे में आपके साथ मुलाकात कराना चाहूँगा और उनके बारे में कुछ बताना चाहूँगा। साथियों, डॉक्टर नरेंद्र इज प्रेजेंटली a faculty member with the Department of Geology in Delhi University. He was born and brought up in Hisar, Haryana. He initiated his academic journey with a bachelor's degree in science in biotechnology from the government postgraduate college at Hisar, Haryana. Driven by his interest in animal biotechnology, he pursued advanced studies, earning master's and doctoral degrees in animal biology from the University of Hyderabad, which is a great central university, very good central university in India. His doctoral program research delved into the understanding of midgut regeneration in lepidotropin pests. His research deals with the examination of extracellular vesicle signaling during wound healing, with a particular focus on cytoskeletal remodeling and its implications and wound response. Try to get this connection with the bioinformatics, wound response, understanding the response, what the wound is speaking, what the wound is needing. Dr. Narendra's work, documented in prominent publications, illuminates the psychological and molecular transformations in insect larvae subjected to continuous sublethal exposure to biopesticides. Bringing a wealth of knowledge to the classroom, Dr. Narendra has taught courses in immunology, human physiology, animal behavior and microscopy and molecular biology. His teaching approach reflects a multidisciplinary perspective, fostering a comprehensive understanding of biological processes. His specialized expertise includes in vivo toxicity analysis, extracellular vesicles, isolations using chromatography, gene knockout and knockdown using CRISPR-Cas9 and RNAi and microinjection techniques. Here the cut and paste technology in biological systems that cannot be done without the bioinformatics systems. Previously, Narinder has taught at the Central University of Punjab during 2020-2022. In addition to his academic roles, Dr. Narinder is currently associated with the Academy of Wound Healing, the Academy of Wound Healing and Skin Regeneration and Society of Extracellular Vesicles. As an active researcher and dedicated educator, Dr. Narinder is committed to advancing the research in the field of zoology, wound healing and diagnostics. His work aims to bridge the gap between research findings and practical applications, contributing significantly to the integration of knowledge in wound healing and diagnostics. So, friends, a young topic with a young man, Dr. Narinder, this is all yours, Dr. Narinder. Welcome.
Thank you so much for the warm welcome, and thank you so much for also discussing about that our work is also in line with the bioinformatics and we cannot do our work without bioinformatics. Indeed, this is true. So, let us get back to the today's topic. We will be discussing about bioinformatics, development and future. Is my slides are visible? Okay.
Yes, your slides are visible. Yes.
Good, thank you. So, the topic, if I talk about the definition of bioinformatics, as sir already discussed, like it starts with the computation. When the computational thing started on race, and in 70s and 90s when there is a boom in the computer processing, similarly at that time the bioinformatics also came in picture. So, advancing our understanding of biology by leveraging computational methods to understand the biological data, to interpret the biological data using computational methods is where we fit our bioinformatics definition. And indeed, as sir already mentioned, yes, it is an interdisciplinary nature of a science. You need all type of awareness. For example, as a biologist you have to be there, computer scientist, also a statistician is also required, and a person who also understand in current era about human physiology or health science. So, all these people collaborate together and work in this interdisciplinary science, and here it comes as a bioinformatics. So, bioinformatics understanding is not alone about computational, no, only about biology, no, only about human health, no, it is an interconnected network using all this data information and then get something out of it. So, what do we actually do in bioinformatics and what we are going to discuss today in bioinformatics? Let me walk through a content of my today's presentation or slides. The rise of bioinformatics is basically with the fueling of lots of data we have. So, what is this data we talk about? It's some kind of a notebook data or something. So, this data is in genomics data, proteomics data and metabolomics data. So, I have bolded these three words: genomics, proteomics and metabolomics. And whenever we have some data, let us say we have some kind of a book, so we need to have a bookshelf to store it. So, this kind of a data which needs to be stored, and also it needs to be analyzed. So, the analysis part also will cover, and also the data is there, but how people will understand it. So, we have to annotate it. So, the annotation part will also be there. Once you have a data, you analyze it, then you annotate it, then what its implication in the biology. So, you have to integrate all of this into a biological network. So, here comes the necessity of storage of all this data, analysis of this data and annotation of the data. Further, if we address these needs, so what are the challenges as a bioinformatician or a person who works in a bioinformatics field, what kind of a challenges they face? So, challenges is like understanding the fundamental of it, because if you talk about the data perspective, we have a lots of data here. I will come to this point, what does this lots of data means here, we will discuss about that as well. Also in the last part of my slides, we will be discussing about how this becomes interdisciplinary and what kind of a training it demands right now. And for the next generation scientist, with this interdisciplinary knowledge, how we are capable to navigate the modern bioinformatics resources and technologies. So these all will be my today's content in our presentation. So let's move to the first part of it, data accumulation. So I have bolded these three words here, genomics, proteomics and metabolomics. So let's get back to them one by one. So first we talk about genomics. So understanding this familiarity that a gene segment will be there in our body, which is in the cell part of the in cell there is a nucleus. This picture which you see here, this is a cell we are showing, and then this is a nucleus which is showcased here. In this nucleus we have lots of genes, and these genes are embedded in this colorful kind of a picture, which is basically a nucleosomes which we are showing, and this is decoded into various number of chromosomes. So in humans we have this 22 plus 1 XY pairs of chromosomes, and these pairs is coded by various number of genes, and these genes are coded into simple 4 factors which is called as ATGC. So these are the code where the whole genome is coded. So the data, if you get the arrangement of this ATGC, so if I start with my session, what is the primary data here is ATGC only. So we only have these 4 codons, nothing else. So this is where the genomics data lies. So all the genomics data we'll discuss about is only ATGC coded, nothing else. Then comes, if we talk about a basic mechanism of genes to protein, then gene makes RNA and RNA to protein. We know that in RNA we have an extra uracil coded added there. So instead of only ATGC, we have AUGC in RNA, that also becomes a kind of a data for us. And then once RNA is formed, then through a cell biology process, through translation, we move into the proteins. So this is a structure which looks like a folded structure to you, this kind of a fluorescent structure. This is a general protein structure. I would not like to name it, but this protein structure is important for doing lots of biological function, lots of interaction, lots of activities in the cell. And when I talk about the cell, then definitely it have an implication on the tissues also, and followed by the overall body. So the next data we have is a protein data. This protein data will be coded by only not ATGC. Now the codes will be changed. So we have a codes for ATG, for example, methionine is coded for ATG. So each triplet codons will be coded by single protein code. So M will be there for protein. Similarly, we will have like UUG, UAC, it's a stop codon. So like this, we have a codon shifting from single codon ATGC to a triplet codon system in a protein. But again, the data which will be dealing here is with respect to the genome, RNA and then proteomics. Similarly, once the proteins are there, genomes is there, now these all proteins are involved into various kind of a chemical processes, and these chemical process involves metabolites, and these metabolites do associate with the protein molecules, and they might act as a small substrate, intermediates or some other functional doing into the cellular process. So this kind of a data if we get, again it's a data form. So if we get this kind of a data, this is called metabolomics. So similarly, so this is the data which were previously people were thinking about only genomics, proteomics and metabolomics, but I would like to tell you that this is not the only data left right now in the bioinformatics. So most of us now wear a smartwatches and other kind of instruments, I'll say. It's just a gear which we wrap it around our body, and we also get an information of how much oxygen partial pressure we have and what is our breathing rate, all those kind of a data also we get. So now the data context is now increased. It's not only genomics, proteomics, metabolomics, it's also about your daily routine workout, daily routine lifestyle. So all those data also comes in a picture. So this is the data we are talking about. So whatever we do, when we do those things, so how we are breathing, what is our heart rate, after eating what is our heart rate came down, so all this also comes in a data picture. Also, if I talk about the, let's say we go to a doctor and we have an X-ray, we have ECG, we have EEG, all those data also come as a data format now. So anything which is relevant to the biological entity will come as a data here, and this is what we have, a data accumulation. And data accumulation means if you collect all those data, then this becomes your primary object to start for a bioinformatics. So the data is very vast. It is not only associated with this genomics, proteomics, metabolomics alone now, it is more than that and it's bigger than that. Now the thing has also changed, because this is one of the slides which represents that we are in 2023, we are celebrating 20th anniversary of Human Genome Project. So those who are not aware, I would like to know that we have started in 1990s and it took 13 years to complete that Human Genome Project, and more than 20-30 institutes, a good funded institute, took this 13 years to complete one Human Genome Project. But just think about it, is it the same thing right now about today? Answer is no. Now in one day a company can easily do that one human genome sequencing very well. So if I want to show you the some kind of a Moore's law, it shows it's a 2001, the cost of an human genome, this is in dollars, so it's like $1 million for one human genome project, but it comes down to $1,000 only. If I try to calculate it in Indian currency, it's around 90 to 80,000 rupees only. So this is how the change has came with respect to the data processing or data acquirement, and this is a report by economist which says that the cost has came down to even less than 1000 rupees. So this is a change we are getting in our data acquisition. If we further move with respect to the number of data increase, so if we see the data cost is decreased, so definitely the number of data will be also increased, right? So if we see the market, this is the market value of a bioinformatics. So global bioinformatics market, according to the CAGR report, it is going to be 38.79 billion in 2030. And if we start right now, it's approx 13 billion. So it is going to be increased more than 15.5%. So definitely there is a lot of scope to work, and there is a lot of demand also with respect to the bioinformatics. Now if we talk about the growth of the data itself, so we will be discussing about various libraries, I will say, where we store our books. So similarly there are various libraries for the genome, various libraries for the proteins. So the user activity and the data is drastically increased from 1990s to 2009. So 1996 where we started talking about genomes, gene bank came in picture in 1993. So from this gene bank we have reached to this 2019 gene bank accumulation, and this is the sequence million reads. What are the sequences? We will definitely discuss. So the sequence million reads actually started from merely 1 to 7 million reads. So the data which we currently have, and let me tell you the good news that most of this data is freely accessible. So anyone who is sitting at the home, they can definitely use this data and work at it. You do not need a very big institute to start the bioinformatics. Data will also come back to that part. So even with your single computer system or moderate functioning computer, you can always start with the data processing and work on the bioinformatics. So let me get back to the part like how the analysis start. Let's say what is this data I am talking about? Definitely the sample can be anything. So I have kept a image where you see lots of animals. So it means even there is some plants maybe missing here, or a virus or a bacteria, anything, any biological entity, you think of it, they will be made up of a cell. Anything which is made up of a cell, if we call it as a living, NCERT living and non-living definition. So whenever we talk about a living thing here, we have a cell. The cell contains now, so I am also including virus here for certain perspectives. So that also have a RNA, proteins and DNA. So this RNA, protein, DNA, we will be collected as a sample source from the various, any organism you name it. Then the collection will be done, has to be done very carefully. You do not have any kind of a mix of or contamination of other sample or other species sample. So let's say if I want to do a sequencing of a parrot, then I should have a sample of parrot only. I should not mix a sample of let's say monkey or other species. So this is what I am talking about collection. So most of us has gone through a COVID era and we have seen the sample collection, and you have seen just by taking a kind of a cotton bud, we are able to collect the sample from our saliva. So this is how a collection happen with respect to the data we are talking about right now. So this is the basic framework of a cellular process. We have a DNA, then we form RNA from it, and then there is a through cellular process we have a protein. So this is a replication, transcription and a translation process. Definitely we read it in our 11th class notebook in a gene chapter, and this is where we talk about isolation. So the isolation can be done for the DNA, the isolation can be done for the RNA, and the isolation can be done for the protein. From the each sample you can also isolate all three of them together and also isolate individually of them together. So the same sample can work for any kind of this biological entity isolation. Now what will happen after this? The isolated content, for example, if it is a DNA, we process it through various kind of a machine. Earlier when we before 1990s we were talking about only Sanger sequencing, but after Sanger sequencing, when the high throughput sequencing came in picture, then we have a lots of data. The data can be processed very easily in a very fast efficient manner. So this is how the step starts, that you have a sample, then you collect it without any contamination, you isolate it, then you have a NGS machines, which we call it right now as an next generation sequencing machines, and followed by this is how we actually process it. So this is a DNA, let's say this is a DNA strand, you actually break it into various small fragments. Why do we break it? I will come to that part also, and then further you again have a short reads of this, and then you align it and assemble it. So this assembly has to be done with respect to the something. The respective perspective here is a annotation. So I'll also discuss about what is annotation here. So assembly means like if you can see me, this is like you break a pen, and but this pen is break and kept apart into various locations, and then you just join it. So this is what assembly means here. So over here you have these kind of a data is called what we call as a raw reads, and we denote this data into some kind of a format like FASTA-Q format here. It's the same like we have an Excel file, so we call it as a XLS file, or if we have a document in a Microsoft Word, we call it as a .doc file. So similarly we have a file reading here as a .FASTA-Q file. So this is a file which we achieve from our initial sequencing. Once we have this file, then we have to check whether the file is authentic or correct or not. Is there any kind of a contamination or not? So then comes the next process, quality control and profiling of these files. So then we check this FASTA-QC file into various kind of FASTA-QC softwares are there by which we do the quality control. We check the overall ATGC, again I am coming back to the ATGC thing. So we will check the ATGC content as well as is there any kind of overlapping of the content, are there multiple reads or not. So all those things come into the quality control. Then we align this all the raw reads against a non-genome. I will come back to this also, what is this non-genome. As we already discussed that human genome is now available, right? We are celebrating 20th it's 20 year now the human genome is being published. So the data is already available. If you get something out of your own human sample, then you can compare this data with the human genome data available online, or again it's a free data available. Then there are other tools which profile it. So what does this profiling means? How do we profile this kind of a data? So the profiling means you annotate it with respect to A versus B. So the colors which you see, this is nothing which we through red color I am showing here, or if you are able to see the green and red color, so assume that the red color is by, like say it's an up regulation, there is one X thing were there, that becomes 5X, or the green you can say X minus 5 it becomes, or the 5 times less it becomes. So this kind of a profiling can be done with respect to the two sample. For example, you have seen in a COVID case, because you have seen the PCR values and you have seen that the delta CT something you have checked, the CT profile. and the content of that particular sequence was higher or lower. Based upon that, you say that there is a viral contamination or not. So similar kind of a downstream application can be done, but not with only one sequence. You have thousands or you have a lakhs of sequence here, and then also you can combine this sequence with the protein data, and then you see how this protein data and genomic data is interrelated. So all these comes under the downstream application of the sequencing. So this is an analysis part. Followed by this, let's say you have a data. So when we talk about the books data, you through the books can be stored into your shelf, like library shelf. So the data which I'm talking about, it's like millions of reads, billions of reads. In terms of a GB content, if there are like 25 million of a read, it might have a 6 GB of a data. So I hope this GB you can understand, 1 GB to, it's a gigabyte process. So this GB of data cannot be stored with you for a long term. And also if you have a storage of this 6 GB data and somebody else wants to work into, let's say, Europe, then how the data will be processed? Either you send it through the emails, so it's not possible. So there are some storage houses, what we call them, or a databases actually we call in terms of bioinformaticians. So these databases are these name as like such, for example NCBI. NCBI is one of the well known database which have all of the, it's a National Library of Medicine by NIH, and this data where we store all our genomic information, protein information, also RNA information. So the library which we are creating, it's not a physical library again, it's a library which requires lots of supercomputers, lots of data processing units. So one of the example is NCBI. So NCBI basically it's in US. So similarly Japan also have its a database kind of a thing which is called as a DDBJ, DNA Data Bank of Japan. Then we have a specifically for the protein, we have a protein data banks. Similarly other is a Swissprot, it's also a data bank for the proteins. Then we have an MBL, this is also a European Molecular Biology Laboratory. This is also a database where we store lots of any of these data kind. Then European Nucleotide Archive. So the short reads which we got as a raw reads also, we try to submit it onto the European Nuclear Archives, and there we submit this data as a profile which is called as SRA profile. Sequence Read Archive means Sequence Read Archive means SRA profile. And then we were talking in the previous slide about the assembly. So if you have an assembly of that sequence, then we call it as a TSA data. So for example, transcriptomic sequence assembly. So we store these files as a TSA files or SRA files and so on. Then we also have a data called string here. So now string is something else. String is not like the data where you have a raw read. String is, as you say, it shows some kind of a interconnection. So string is a database which shows you a protein-protein interaction among living organisms and within the living organisms. And this database showcase you like this X protein is interacting with Y protein or not. So if you have a protein and if you want to check whether the protein have any kind of an interaction or not, then you can just go to this database, just type your protein and you can see that. Similarly, we have a KEGG pathway analysis. So it's a Kyoto Encyclopedia of Genes and Genome. This is a KEGG pathway. So over here also we have a database where you see lots of pathways being recorded. So all these storage house has a specific information to store, but one good thing to tell you here that all these storage house also interconnected. It's not like you will only find the information on PDB. There will be some information which will be available on string, then PDB protein data will also have a link for the string. So similarly all these storage houses or databases also interlink, though they have a separate data storage also. Coming back to the annotation, what is this annotation? So to rephrase the annotation well, I have kept a poem, and this poem reads like two roads diverged in a yellow wood, and sorry I could not travel both, and be one traveler long I stood, and looked down one as far as I could to where it bent in the undergrowth. So I was actually reading this poem because I am not well aware of the poem, but if I try to annotate it, so if I say a yellow wood, so it means it's a autumn, okay, and if I say through the poem I saying I was looking it down, so it means you might feel regretful. You said a sorry is there, so it means regretful. Look down one far as I could shows, it says that there are time to think. When you are looking down, then you are thinking something. Maybe you are looking up or down, the both way you are thinking. So means there is some kind of a words are there which kept by the author or the poet, and then you try to decode it. So when you have these kind of a thing, then it is called as annotation, annotating something. So similarly, we had ATGC as a component here, or a MSRUV as a component for the protein. So when you try to decode this protein or DNA function, location, that becomes annotation for the genomics, proteomics and metabolomics. So process of identifying the ATGC, I'll say, or protein, will stick to the ATGC perspective. So when you try to identify the ATGC location, what is its function, what is doing in the DNA sequence, that comes the genomic annotation or gene annotation. So identification of a various features, when you talk about within the genome sequence, where its start points, where its end point, we'll talk about that, what is the start point and end point also. What is an exon and intron? I hope you are well aware of with the exon and intron, again NCERT. 5th chapter where we talk about the genes. So exons are the essential parts and introns are the non-essential. So this is how we know the sequence is essential or not. Then what are the regulatory reasons, how this sequence have a higher profile of expression or a lower profile of expression. So the regulatory reasons where the promoter is binding. So again, what is the promoter? I will also talk about it. So this whole area, when you try to figure out what this ATGC actually means to you, what this ATGC combination means to you, because there is again ATGC, but I am discussing in terms of billions and millions. So ATGC will be in combination of this million in various type of sequences to form this millions and billions of reads. So try to locate them, try to identify them, that's what comes as an annotation. So we have a protein annotation, gene annotation like this. So over here I kept a bar. This bar actually represents as a DNA slide or a DNA fragment, and there is a protein which I talk about, an RNA polymerase. So RNA polymerase binds to this area which we call it as a promoter region of a DNA. From here, once the RNA polymerase binds, so it actually tells us that this can now synthesize an RNA. So we have a replication, transcription and translation process. The process I am showing here is a transcription. So transcription is a process, means out of, let's say, assume lakhs of the coding sequence, there is a one fragment which has to turn into the RNA. So if I have to turn an RNA, not out of that lakh sequence, I only need 1000 base pairs of that. So we call it as a base pair. Base means single A is a base. So base pairs we call it. So this definitely have a start site. There is some start site has to be there where you start the RNA formation. Then you will have a start codon also. There will be a specific region, will be a coding sequence region, and then you have a stop site also, because out of that lakh or thousands of base pairs you only need 100 base pair assume. So you only need this kind of a fragment. So when you annotate this kind of a structure into this ATGC, GTGG, ATGC kind of a fragment, this becomes annotation. A similar annotation can be replicated, let's say for this is for RNA, when the transcription happens. So you have an U coded here. So as I was mentioning earlier that this is in DNA codon, it's in a duplicate we have shown. You have a double sided fragment, and then you have an AUG, the T will be here replaced with the U, and this is how once the translation happen, the coding will be again converted to some other code format which is for protein. For example, CAU is code for H, CUG for L, it's leucine, tryptophan, phenylalanine. Like this we have a multiple coding system for the translation always. So this is again a coding, and I believe if you are sitting at home, so doing this 20 or 30 base pairs of a coding is very easy, right? It looks very easy, but when it comes to lakhs of base pairs and then you have to code it, think about it. Can you do it manually? No, it's not possible. So to do it, definitely we need a computational biology here, or computation let's say, then biology perspective comes later. So computer is required which have a lots of calculation ability, higher processing speed like this. Other than this, this is about an example of a DNA, we talk about a DNA RNA coding. Similarly, we have another coding methods where we code for the protein. So these all structures which you see, these are the proteins. So over here, this is a nucleosome protein, potassium channel protein, some thrombin protein. So all of these are different structure of a protein. So this structure this, if I consider this as a cellular membrane, so this particular protein is like half of the way of the cellular membrane, maybe outside of it, and this may be inside of the membrane, and this may be inside to the cytoplasm but towards touching to the inner part of the membrane. So how do you know like what part of the protein is touching to the membrane? Out of this you see some blue color, or some, let's say over here is a blue color and this is a pink color. So these are, let's say, two subunits of a protein. Which part is interacting with each other? How do you know that? So all these parts, which is an interacting part, which is a subunit, is going through the cellular membrane, which is going through the outside. So all these can be used for the annotation. So even after annotation you can figure out, let's say if you compare our genome, how do we know that orangutans are the closest one or gibbons are the closest one? Why not the lemurs are the closest one for the human? So how do you understand this biology? Again, just by simple comparing to them. Then you also can compare even a single protein. So there is one of the protein, one of the largest known protein of human is the dystrophin. It is also known for the muscular dystrophy. So dystrophin, how do you assess that for humans a crab or a fish or a cephalopods will be a closest model system. So all these understanding can be come from the bioinformatics bioinformatics data. There is another perspective of the bioinformatics when we talk about the drug discovery. So recently you have came across for the drug development for again COVID or Covaxin. How the Covaxin, so you must have read that few companies, I would not like to name those companies here, but few companies have used a similar drug which has been used for the malaria. And the same drug can be used for the COVID. This kind of a news you have kept hearing. And even after that, there are thousands and lakhs of molecules, biological molecules, how the specific molecule is being used. So the first work which is I have shown here, preclinical studies, where a scientist sits with an objective that he has to find a solution for that particular drug or particular disease, then he tried to figure out a novel chemicals. These novel chemicals can be synthesized and also can be computationally modeled. So all these data, when you try to model a new chemical and try to look upon whether it could be useful for the disease or not, all this work can, earlier it was actually taking around, let's say, I'll not try to exaggerate it, but it's like 3 years, 2 years, 4 years. But now a good bioinformatician can do this work within a 1 week or a 1 month, depending upon the type of during the COVID era, this has been shortened a very well time frame. Then the rest of the process goes same for the drug discovery. You use this chemical, you do a preclinical test on the mouse, and then the formulation comes up. The same process, then how it comes to the company, this remains same, but this took a lot of time earlier. But this time we have decreased to merely weeks, and definitely we will talk about the AI also, artificial intelligence, which has actually decreased the time very less. So what is this artificial intelligence I am talking about here, and how it comes to in bioinformatics perspective? The future which I was to talk about in bioinformatics is something is related to the artificial intelligence. So what is artificial intelligence? So simulation of human intelligence into machine that are programmed to think and learn just like humans. Again, I am saying it's a simulation. You train a machine to think like us. It will not train itself. Also, it will train itself in a different perspective that I will let you know also. So this simulation is where we talk about artificial intelligence, and again it involves a development of various algorithm and a computational models. So it again requires a computer. Okay, so there the work which were done by traditionally for the human intelligence, let's say we were talking about this CAG or codon understanding or codon annotation, this can be also done by AI, because the AI we can train or program the AI for this, and the AI can do this work. Now AI encompasses various kind of a range for the techniques and technology, in which include machine learning, natural language processing, and computer vision and robotics. So what are these? We'll just get a brief touch on this. So this is how AI can be used. So what is this machine learning? Like you have a sample data and it is processed into your computer, and the computer recognize the pattern of this data and make an algorithm by itself. This is what we call it as machine learning. So there is always some data is there. So let's say human genome data is already available, then you use the human genome data through machine learning and you give a random sequence of ATGC random sequence, then the by machine learning it will tell you, okay, this particular sequence is coded for the, let's say, insulin protein. So like this the machine learning can work. Then again this computer system can also work like a neurons like it is being interconnected to each other. So this is where we talk about neural networks in artificial intelligence. You can talk about the X protein is related to Y protein and interacting with the next protein. So this interaction among the protein can also be done using neural networks. Then we have something called natural language processing. So this is something like when I hear something, I learn that particular thing and then process it and analyze the document. Let's say we do it in our classes, we hear what our teacher says, and whatever we hear, then we try to decode it into our books. He said us some story, then we read this story again and then we decode it ourselves. So this ability of natural learning process can also be done using artificial intelligence, and robotics is very well known now, but how artificial intelligence can be used here, like you have a robotic surgeries available. So machine can assist people without the involvement of the people. So this is how a robotic artificial intelligence can be there. So bioinformatics, how we can fit the bioinformatics into AI. So because we have a lots of data available and the data is this much very high, as I was showing you the pictures, it's in like we have a 37 billion reads, even into for even a single insects. If I worked with the insects as Gagan sir was mentioning again earlier. So insect RNA data, only the RNA data, it for around 32 million sequences, we have only let's say we can take it into 6 GB data. So 32 million reads into 6 GB data. This is only about one single species of an it's a Castor semiluper, one of the Lepidopteran species. So that single species have this much of a data. Now think about you have a let's say 10 number of a species or 20 number of a species and you want to compare that. So how to deal with this big amount of a data? Again, even if you sit with the higher configuration of a computer, again it becomes a copious data, you can't handle it. Also you will miss lot of information out of it. So the prediction which you actually have, you could have done better, the prediction is again missing from this. So this AI can revolutionize this technique. The future lies here. You use this big amount of a data and enhance your approach for predicting the annotation better. So there we can use AI information in the bioinformatics. Then if we talk about, let's take one example how this actually machine learning works. So machine learning, let's say you have there are various kind of a algorithm. I hope you are well aware with the trigonometric equations. Similar kind of a equation with the mathematical modeling we apply here. So one of the supervised learning in a machine learning is like SVM theorem, regression theorem, random forest theorem. I will not go into the detail of this, but just to give an idea, a regression can be used for the genome-wide association analysis. A random forest theorem can be used of pathway based classification analysis. A CNN network analysis or a deep learning can be used for the protein secondary structure prediction. And this has increased our output a lot. I will just come on to that part also. So this kind of a machine learning come into a supervised learning machine learning. The other one we have is unsupervised learning, where you cluster the data. So this kind of a clustering comes in the hierarchy of a data, and we can cluster the protein type. This protein A has a subunit 1, 2, 3, like so we have a protein family subunits. Let's say you have various kind of outliers in the protein. So this gene expression outliers can also be taken out by a PC analysis. So this whole data comes into a dimensionality reduction. So you have a lax of gene, but you are focusing on one type of a gene, but you do not have that prediction capacity. So over here machine learning, due to its dimension reduction theorems, it can help you to reduce those outliers which should not come into your data. Also this can also help you to fix some boxes for you. You might have some missing data. So missing data can also be taken up from the machine learning perspective. So okay, let's say this machine learning again helped in some way. So why can't we know that in what perspective and in what range? So just take an example. This is a protein structure of some molecule, and trust me, getting a protein structure folded this fold like this, it's not easy. And I hope all of you are aware with the Professor Ramachandran work with the Ramachandran plot, where we talk about beta sheet, alpha helix structure. So there also we only discuss about protein structure, but getting the structure is not easy. So now you can ask me, sir, that this can be done by computational. Answer is yes, but at what speed, and at what speed we have increased this structure formation or structure prediction. Let's say I give you a sequence of ATGC, 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 and then you have to predict a structure out of it. So how you can do it? So there are various software to do it, but is the speed of the software has increased after the future which we are talking about, machine learning and AI? Answer is yes, the speed has increased. Let's say there are these various conformation, and these conformation, a slight change in a protein can leads to a various effect. So protein structure conformation is very important, and this is the accuracy we have achieved. So earlier in 2006, when the AI was not talked well, so that time the protein structure accuracy was around 37%, which has we earlier in 2018 when I was a PhD student, we used to get the structure confirmation or accuracy possibility around 57%, but today's the data now reached to 87%. This is one of the software AlphaFold which we use in our daily routine analysis for the protein production, protein prediction here, and the prediction is now accurate to 87% because you have to know the exact prediction time. So now let's try to end this topic, and the question comes, how do we start it, like where we can start it. So as I was showing last picture of mine, that you just need to have one single computer, a desk of yourself, and sit quietly and think what problem you want to address. If you know the problem, that this particular sequence I would like to know the protein data, protein structure of it, or whether this protein is involved into some kind of a disease or not. So if you have a problem problem with you, then try to address it with just a simple mathematical tools based upon the online data. Again, the data which you need to have, you do not need a very supercomputer initially. The data can be processed online server, so you just need an internet connection, a computer or a laptop, sit pet and start your work of a bioinformatics. So thank you for listening me, and I'll end the thing. In future, the huge data which we are getting day by day, this has to be this has to be dealt with using AI, which can save our time and resources, and this can definitely accelerate the biological discovery, especially in the biomedical fields and also robotic surgery. So there are various things you can think you can apply using AI tools and bioinformatics. So this is a brief about bioinformatics. Over to the Gagan sir.
That's great. Colleagues, friends, we have been talking about tailoring of materials in this series and outside this series, and I am basically a student of physics. You know that I have been saying that tomorrow's the days are like that if you come to me and say that I want a material which has so much of material strength, so much of Young's modulus, so much of thermal conductivity, so much of electrical conductivity, those materials are not presently available in nature, but not have been presently made, and probably after 20 years or so we will be able to do we will be able to tailor the materials the way we want it. So I am very happy to learn it today, that in the case of medical science as well, in the case of the cell structures as well. I remember Abhishek Mishra's session in this series last year, where we talked about CRISPR technology and the Carpenter's Nobel Prize.
Emmanuel and Carpenter, right?
Yes, that Nobel Prize where and Sohitra Sarkar, we made these observations about this, that how the tailoring in biosamples can also be made. I am very happy to see. Can I come back to very quickly to a slide, with probably the 5th slide or so. Can I come back very quickly?
Sure.
Where you are showing the cost cutting with the and the time savings with the years, how it has progressed. Yes, the second part, the graph you showed, that Yes, in 1990 when it started, that it was started with the 13 years genome project, which all of us know. I mean, it was a
huge success,
and huge stories in all the newspapers, televisions, etc. And it was indeed, as I remember, though I am a student of physics, but I still remember that has stolen the whole networks at that time. And now you said that it can take one day. And I remember Benjamin came here from Israel, who got the Nobel Prize in 2007. Probably I may be missing the Jewish Israeli scientist. He said that finally it may come down to 70 minutes.
Yeah,
it may come down to.
Yes.
So I think that particular kind of expectation. So 15 years have passed since then, when the Benjamin got the prize. See, after that, now comes some ethical kind of issues in it. See, when you are able to decode the genome like this, what do you think that it is? Is it not something like an astrologer who is a perfect astrologer and says that this is what is going to happen with you after a few years? And similarly, if a medical scientist or if a medical practitioner tells it that look, at the age of 60, you are going to suffer by this disease. So what did we turn into that, to knowing the person at the age of 10 that he will be having that kind of disease at the age of 60? Okay. So are you really helping, or really
no. Okay, I'll put it into this perspective. So the medical help we have right now. So whenever you have some kind of a disease, then you go to a doctor. The doctor prescribe you a medicine. X or Y medicine, then you get treated. But the healthcare system don't have to be like this anymore. It is changed now. You are wearing lots of monitoring gadgets with you, so you can monitor your, let's say, even partial pressure of oxygen or your heartbeat. So within this monitoring, the system can, maybe in future, the system can tells you, okay, now you tend to get this kind of aberration in your system. And then before getting the disease, you can start taking the precaution. So it's like our system. Instead of waiting for the disease and get treated, it's better you should get aware of it.
So you expect that it is
soon, and it's already
that
available.
The treatment would be available for all such kind of disorders.
For an example, if I go to any kind of any company store, Samsung, Apple, or any big mobile store, you see one section, you will see a wearable section. We will see the gadget will have a health monitoring device.
Yes.
So definitely the health monitoring system has to be turned into a different perspective. It has to be a predetermination of these factors. Then you can say, okay, you don't have to wait for the disease to kill you. You can assess it earlier and then plan for the better treatment. So this era, soon it will be coming.
So I'm sorry that though I asked Tanvi to start the discussion, but looking into your last slide, I was so tempted. But this makes me to feel a little more that the emphasis of medical science as we see today is not so much towards the longevity of life, rather than the better upkeeping of life. आप जितने भी रहो, अभी आशीर्वाद भी देते हैं ना, तो भाई लंबी जिंदगी तो हो, लेकिन लंबी जिंदगी के साथ जितनी भी जिंदगी है उतनी सुखमय हो, हेल्थ वाली हो. So better health is a
obviously better health is a better perspective first of all.
Yes. So Tanvi, I stole your time. I'm sorry. Please.
No issues, sir. Your question was more relevant.
No, sir, please.
So when we are talking about bioinformatics and genes and genomics, I could think of this incident which is very common these days. As in, this incident is in newspapers every day. So there's this millionaire. He's 46-year-old and he's in California, and he's taking some pills, 111 pills every day to be younger, 18-year-old. So my question here is this even possible, or is he a guinea pig for some experiment?
Okay, so
a little difficult question for a bioinformatics person, but anyway.
Thanks to that I'm not purely, I'm like mix of both. Again, I've done my biology well.
Yes.
My teachers were good. So the thing about taking pills and making itself a guinea pig. So I remember when doctors said, like, instead of being in a pain, it's better to take one medicine. So in that perspective, if I think that he may not be a guinea pig, he have some kind of a disease and he is taking cure of that. So that is the answer to for take a pill or not. Now the next part you have asked, like thousands of medicines, like what will do the thousands of medicines, right?
Will he become younger with this procedure?
So every other day there is one thing which we talk about in biology is telomere shortening. So as we age, the telomere shortening happens, and if you just type on the Google telomere shortening research, there are lots of research people working on longevity. Definitely they are planning, or they are working at least, how telomere shortening can be reduced. So skin regeneration, all these are coming in a picture. I can't say because I'm also in my 30s, so it will be very difficult to answer. Can we achieve this? May or may not be, but yes. But overall, the process comes under your own consciousness. It's you have a monitoring system, fine, but body knows what it needs, what it does not. So it will also have an implication. So taking 1,000 odd pills sometime may help you and sometime may not be. It depends upon individual to individual.
Yes, Narendra, we remember Michael Jackson's case where he has undergone several surgeries just for the same reason. Actually, our situation is, our children's situation is that we want to promote कि वो सपने देखें, वो आँख खोल करके सपने देखें. और तन्वी's question was very pertinent to this. कि सपने देखो भाई, आगे देखो क्या होता है क्या नहीं होता है.
That's why I kept the last picture only. Even a student who is just have an understanding of using internet, have some kind of a curiosity to know about a protein or a sequence, they can always Google it and go to online, and it's freely available. We have an excess of it. So yes, you can dream it and dream bigger.
Thank you so much, sir. Thank you for your time. Thank you for presenting such a beautiful presentation on bioinformatics, and you talked about not just development but its future as well, like you said. So thank you so much for being with us today. It was wonderful having you, and we definitely have a lot of questions, but because of the lack of time, we cannot raise many. And thank you on behalf of our entire team for being with us today. Thank you.
Thank you so much.
Thank you, Gagan sir, for bringing him along.
Yeah, thank you. Friends, after having seen or having been a part of today's exciting session with a young man, Narendra, on a young topic, relatively young topic, we are coming back to you on 22nd of December with another exciting topic when we are going to have Kuntal A. Joshar with us in our studios. Kuntal is the first vegan person who scaled the Mount Everest in May 2018, and coincidentally friends, the first globally first vegan woman who completed the scaling of Mount Everest is again an Indian, that is Prakriti Varshney. Though the first woman who climbed the Mount Everest in 2022 or so something, not only 1975, Junko Tabai, Japanese. She was a vegan person and she climbed the Mount Everest, but she could not complete the full expedition. She lost somewhere in between while coming back from Mount Kuntal. So Kuntal was 2018, and then 2022 to Prakriti Varshney were the first vegan persons, male, and Kuntal was the first person, and Prakriti is the first woman who completed the scaling of Mount Everest. So Kuntal will be here with lot of excitement. He is still a mountaineer and he is now a freelancer in Mumbai and associated with many organizations across the world, a brand ambassador of many mountaineering institutions. He will be coming with us with a very excitement. We are really looking forward listening to Kuntal here in our studios.
Absolutely, sir. This is going to be very exciting. Moving mountains with Ahimsa with Kuntal, and I think I am definitely going to have a lot of questions, because all the youngsters these days, they have so many dreams and they love to climb mountains, not just doing expeditions, but even for fun they love to do that. So this is going to be exciting. Thank you so much, sir, for the information.
Kuntal will be a great fun, I believe.
Absolutely. Yes. Thank you so much to the entire team and to all the viewers for being with us today. I really hope that you enjoyed this today's program. और अगर आपके पास कोई भी सवाल है, कुछ पूछना चाहते हैं, तो आप हमसे पूछ सकते हैं. और sir has something to say.
Sir, I just missed, I am sorry, I just missed to acknowledge the open sources which were used for taking the diagrams in today's session. We thank Mutle, Food, Nature, etc., from where we had Narendra has taken the illustrations. We thank them. We acknowledge their contributions and thank them. Sorry.
Absolutely, so credits are equally important.
Yes.
Thank you to all the viewers once again, and next Friday on 22nd, just like Dr. Gagan Gupta announced, we'll come back here sharp at 10 o'clock and we'll be talking to Kuntal. So please be ready with your questions, and if in case you have missed watching today's program, you can watch it on NCERT Official, that is our YouTube channel. Please keep on watching eVidya channels. We have a lot many programs for all of you in the day, and they are definitely going to help you in some or the other way. So with this, we are wrapping up today's program, and I really hope that you have a great day ahead, and please be happy, be healthy, and stay with us every time. Thank you so much. I'm Tanvi Khurana. I'll take a leave of you. Namaskar.